This video will discuss the ethical issues in modifying human genes using a method called CRISPR-Cas9. The human body is consistent with 20,000 genes and over 2 billion letters of DNA. DNA consists of two strands twisted into a double helix held together by a simple pairing rule. A pairs with T, G pairs with C. The CRISPR method is based on a naturally occurring system used by bacteria to protect themselves from infection by virus. When bacteria discover the presence of virus DNA, it produces two types of short RNA. One RNA matches the invading virus and is called a guide RNA. These two RNA form a complex with a protein called Cas9. Cas9 is a type of enzyme that can cut through DNA, also known as a nuclease. When the guide RNA finds its target within the viral genome, Cas9 cuts the viral DNA, thereby disabling the viral genome. Scientists realize that this process can be applied to any DNA sequencing at a precise location by simply modifying the guide RNA to match the target DNA. Once inside the nucleus, the resulting complex will lock onto a short sequence known as PAN. The DNA template pairs up with cut ends, recombining and modifying the original sequence. Now we will learn the applications of CRISPR-Cas9.
Here are a few of the functions that CRISPR can be engineered to do. Activate or silence particular genes through interference or DNA methylation, edit the DNA of somatic or body cells, or introduce DNA into an organism's germline. In a research article from the Journal of Clinical Research and Bioethics about the ethical issue using the CRISPR-Cas9 system, Dr. Eduardo Rodriguez outlines the following application of CRISPR-Cas9. Create animal models for research, to control invasive species or pass-through genes, to produce GMO crops more resistant to disease and pests, to cure diseases by replacing malfunctioning genes with working ones, to facilitate transplanting animal organs into people by eliminating retroviral DNA, and to alter DNA in human embryos. These applications have many ethical implications to consider when using CRISPR-Cas9 in practice. There are many unknowns for the future and many boundaries to set for gene editing. It becomes cheaper, faster, and easier for scientists. Here are a couple of ethical problems or questions to consider for the CRISPR-Cas9 system.
1. Ecological disequilibrium. How will genetically modified organisms affect the environment? A gene drive introduced into the environment could be transferred to other species or mixed with other genes, forever changing the ecosystem. What are the possible unintended consequences of editing a genome with CRISPR? Off-target effects or edits in the wrong place with CRISPR could cause disastrous effects for the genetically modified organisms. Once a gene drive is released into a population, how can we control it once something goes wrong? Controlling the natural spread of a gene drive within a population is nearly impossible. 2. Regulation. How will the Food and Drug Administration handle the expanded market of genetically modified organisms as a result of CRISPR-Cas9? Who has the right to organisms that are genetically modified using CRISPR technology? Should biotechnology companies be able to patent CRISPR-Cas9 for the use in humans? What should the regulation look like for CRISPR-Cas9 in the use for humans?
Until recently, all therapeutic cases of CRISPR have been in human somatic cells, which have been deemed ethical using a cost-benefit analysis and informed consent. However, for germline intervention, there are problems with informed consent, since the genetic changes can be transferred through multiple generations. We must consider several important questions here. Firstly, what are the unintended consequences or risks of genetically modifying human embryos? Secondly, where should we draw the line for manipulating the genes of human embryos? Should a parent be allowed to select the features of their child? And lastly, how does the concept of designer babies go with popular religious and moral beliefs in society? Who will have access to genome editing technology? Will the technology be equally accessible in all regions? And how will gene editing technology further widen social disparities? For instance, the technology can be used to enhance an intellectual capacity or physique, causing some individuals to have a genetic advantage over others. These are all questions to be considered before CRISPR can be applied and practiced. Now let's see what an expert has to say about this topic.
I have thought a lot about the way that the CRISPR technology might be used, and I mostly feel excited because I think that it's a very exciting technology that's going to do a lot of good in human society and for human health. I do think that there are reasons to be cautious in the way we implement it for various things, in particular for doing clinical applications in embryos, like human embryos. If you do the editing in adult tissues, those are changes that are made in an individual, but they're not passed on to children. I think most people would feel that genome editing in adults is, or at least might be, for some applications, a very appropriate technology. It might be analogous to taking a pill or any other kind of therapeutic drug that you might use in a patient for treating cancer or some other disease. And if you could do something profound like eliminate sickle cell anemia or eliminate cystic fibrosis, or have someone not have to suffer from Huntington's disease anymore or worry about getting it when they get older. What certainly raises ethical implications is whether that sort of change should be made in eggs or sperm or embryos, where those changes could then be passed on to the next generation.
So what do you think of the CRISPR-Cas9 technology? Should it be allowed? Is its usage ethical? These are all questions we must ask ourselves, not just for this, but for any technology before its mass usage.